Okay, as promised, here's your tutorial for culturing a sample of bacteria from your pGLO Plus plate. Now, before you begin, just in case you didn't get a chance, you need to assemble your supplies. So from your general laboratory supplies bag, you need your large test tube rack, your tube of rubbing alcohol, your Sharpie marker, and you need your microtube rack. From your activity number 9 bag, you need the tubes that are wrapped in orange tape that are labeled LB plus amp, and you'll need a sterile loop. In addition, you'll need your your plate of pGLO plus bacteria. Okay. The bacteria themselves, you can tell they're bacteria because they have a slightly sort of off-white color and they form teeny tiny little dots on your plate. So if you see anything fuzzy, that's probably mold and not your pGLO plus containing bacteria. And if you see anything orange or anything with a different color, that's probably a contaminating bacteria. Okay. So the first thing you want to do is unwrap your microtubes of LB with ampicillin added to them. Now we add ampicillin in order to select for the growth of bacteria that contain our pGLO plasmid. If you'll remember from Tuesday, the pGLO plasmid is actually engineered to carry 2 different genes which code for 2 different traits. One of those genes is the green fluorescent protein gene, GFP for short. That gene, when introduced into an organism, provides the trait of glowing green under UV light. Okay, so that's why when you shine a UV flashlight onto the cells from your pGLO plus plate, they should hopefully glow green. The other trait that is coded for by a gene in the pGLO plasmid is an ampicillin resistance gene. So any cell that contains a copy of the pGLO plasmid also gains the ability to grow in the presence of ampicillin, which is an antibiotic which is normally used to kill bacteria. Okay? So we add ampicillin to our growth media in order to select for the growth of bacterial cells that contain plasmids. Any cell that loses its plasmid or never took up a plasmid, these cells should lack an antibiotic resistance gene and they should not grow. Okay? So anything that grows on our media containing ampicillin should contain our pGLO plasmid. Okay, so the first thing we want to do when we grow up our bacteria is we want to take our tube of rubbing alcohol and use it to clean our loop. So we can take our loop, we can dip the loop into the rubbing alcohol, swirl it around a second, and then kind of wipe the extra alcohol off on the side of the tube. Then we can give this a little shake until it dries. Okay, now my loop is dry. Now I'm going to take my plate of plus pGLO bacteria. And the first thing I'm going to do is I'm going to look at the plate and I'm going to identify the bacterial colonies, okay, which is a term used to describe the masses of cells, okay, on the plate. Okay, so each little individual dot that you might see on the plate is actually a colony, and it contains millions of cells. Okay, and all of those cells arose from one single parental cell. Okay. So, the bacteria that contain pGLO to our naked eye should look like teeny tiny little off-white dots. Okay, and you can hopefully see them on my plate here. Okay. If you see anything fuzzy, that's a mold, that's not a bacteria. Okay. So I'm going to take my sterile loop and I'm going to pick up a sample of one of those colonies. Once I pick up a sample of the colony, I'm going to take one of the tubes of LB with ampicillin, okay, and this is just liquid media. It has the same components as the stuff that's in our plates, except this stuff is still in liquid form. This stuff we add agar to it to make it solid. Okay. So I'm going to open up one of those tubes. I'm going to dip my loop into the tube, and I'm going to swirl it around to drop a few of those bacterial cells into the liquid. And when I'm done, I'm going to close that tube. Okay. This I'm going to put into my rubbing alcohol, just so I don't have to hold it. And now I'm going to take my Sharpie marker, and to that tube that I just added sample of cells from my plus pGLO plate, I'm going to label that tube plus pGLO. And I'll just write on the side of the tube in order to label it. So we're going to do one more tube, so we'll have two tubes that we're culturing bacteria in. Now we actually gave you 3 tubes of LB plus AMP. One of those is just an emergency backup in case maybe one gets knocked over and spilled. Okay. So we might have one extra tube, and that's totally okay. So I'm going to take one of those tubes, you have 2 remaining, we're going to take one more and we're going to add bacteria to it. So again, I'm going to take my loop out of the alcohol. And if you find that there are any chunks of bacteria on the loop, you can kind of knock them against the side to get rid of those chunks. Okay. Dip my tube loop in the alcohol and kind of wipe the alcohol off the loop, okay, just like this. And then I'll shake it again to let it air dry. Now that I have a dry loop, again I'm going to take my plate of bacteria and I'm going to use my loop to just pick up a sample of a colony. And then I'll take that tube, open it up, and just shake my loop in there. Just knocking a few cells off the loop into the bacterial media. I'll close that tube when I'm done. I'm just going to store my loop in the alcohol. And I'm going to take my marker now, and I'm going to label this tube pGLO plus as well, because I added a sample of the plus pGLO bacteria. And I'll put that in my rack. So, that's it. I should have 2 tubes that I've added pGLO plus bacteria to. Now what we'll do is we'll take these tubes and we're going to incubate them somewhere warm to give the bacteria time to grow. So hopefully by tomorrow, as those bacteria grow and reproduce, the media is going to get cloudy. And we'll go from having maybe 100 cells in each tube to maybe having millions. And as those bacteria reproduce, they're going to copy the pGLO plasmid inside of them and pass that plasmid and the traits that that plasmid encodes down to the offspring cells, which in biology we call daughter cells. Okay. So, to make sure that these guys grow efficiently, we want to keep them warm. There's a couple of ways we can keep them warm. We can use our heat block. So if your heat block can maintain a temperature of somewhere between 30 and 37 degrees Celsius, we can put our tubes in the heat block. Okay, so for example, my heat block is set to 35 degrees Celsius, but it seems to be holding temperature at around 36 and a half. That's just fine because that temperature is between 30 and 37. So I'll use my heat block. If your heat block isn't working, that's okay too. We have a biohack for you. You can take these tubes and you can keep them warm by putting them in your pocket. Now you want to keep those tubes in your pocket all day until you go to bed. When you go to bed, you can take the tubes, put them in your tube rack, and just leave them at whatever the temperature your house is at. Okay. If you don't have pockets in the pants you're wearing, or you don't want to walk around with tubes in your pockets all day, you can take your tubes, you can put them in your rack. You can go find a window that gets a lot of sunshine, or maybe you have an enclosed porch that gets warm, or a greenhouse. And you can take this and you can put that in that location. So I might put this on a windowsill that gets a lot of sun. And I'll just leave those there until tomorrow. So no matter what heat source you use, your heat block, your pocket, or your windowsill, we're going to leave those cells to grow all alone until tomorrow. As for cleanup, we can throw away our loop. We don't need this anymore. Let's hold on to our pGLO Plus plate for another day. So you can take this and you can put it back in your Go Stem kit. We'll save our tube of rubbing alcohol. We'll need this again possibly. So we'll put this back in our general laboratory supplies bag. We'll put our tube rack back in that bag, as well as our microtube rack and our Sharpie. This is going to go in the garbage. And this I'm going to stick back in my Go Stem kit. And the rest of activity number 9 bag, we're going to save and we're going to use tomorrow. So that's your tutorial. Good luck, and I hope it goes well.